Hello friends, good afternoon and welcome to EduSat live lectures. Dear friends, today in immunology we will be talking about immunological methods. To discuss this topic we have with us our subject expert Dr. Anita Kamra Verma. Dr. Verma is Associate Professor in Department of Zoology in Kirori College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you, Amrit. Good evening. My very dear students, today we will talk about immunological methods. And as from the beginning, I have been saying that immunology depends on only two things, antigens and antibodies. The reaction to anything foreign which goes into the body is the response, and it is called as the immunological response. So obviously, whatever has to be detected, there has to be a certain methodology by way of which we have to detect. So how to detect the antigens or how to detect the antibodies will form the basis of today's lecture. Immunological methods are numerous, and I would be dealing a few to begin with today, mostly serological tests. So to begin with, what am I talking about? I'm talking about antigen-antibody reactions. And why do I want to perform them? Because I want to see what are the titer levels. That means what is the level of antibodies being raised against a particular antigen which has entered the body. Like I had explained earlier, anything which enters the body, foreign, living or non-living, is an antigen. But all the antigens may or may not be an immunogen. Immunogen is a particle, living, non-living, which elicits an immune response. So when I do any test, if I perform a test to elicit the amount or the quantitation, whether by visualization or whether by a colored reaction, I would call it as immunological method. So the easiest is to do a serological test. It has to have two components, one antigen and the other is the antibody. Whether the antigen is known and we have to detect an unknown antibody, or we have an antibody and we have to detect an unknown antigen, depends on what type of experiment we want to set up to visualize this or to just quantitatively have a colored reaction. So when I look at a cartoon, what do I see? That this is a diagnostic method. So I say immunodiagnostic techniques, where I say it is directly visible. That means it could be a precipitation reaction, it could be agglutination, or I have a range of other techniques which tells me how to detect by immunofluorescence, by radioimmunoassay, ELISA, which is an enzyme-linked immunoassay, or by flow cytometry, chemiluminescence, etc., etc. So when I talk of a serological test, what do I test? I test the blood. So it is very easy to take blood from a patient and then separate the blood serum from the RBCs and other particulate cells. Then if I take the particulate matter, what is a particulate matter? That would be the bacteria or maybe the RBCs. But when I say the soluble antigen, then that becomes the antibody which is present in the serum. So for a serological test, I do two things. I remove the blood clotting proteins and I just take the serum, or I take the non-coagulated blood and we take the plasma and start doing the test. So when I have seen a test with latex beads, this is a very common pregnancy test. This coated with an antigen, when reacted with the blood, gives us a test of negative or positive. That means here what I require is the antibody to be divalent. I have a monoclonal antibody, I have polyclonal antibodies. What I require for any reaction is that the antibody be divalent and it cross-links with a multivalent antigen to form a lattice network or clumps. That is what is called as agglutination. This is a very common reaction where we see the blood groups are based on such kind of reactions. When I say agglutination reaction, I can have a positive result or a negative result. So that means if the lattice is formed along with the antigen and the antibody, what I see that there is an agglutination, this will show a positive result and this will show a negative result. Then I am saying agglutination, which I will carry forward to something called as hemagglutination, which categorically tells me that this is involving the red blood cells. So even a viral infection can clump the RBCs, and this will be called as active hemagglutination. That means the antigens from the virus will directly cause the RBCs to lyse. But this can easily be inhibited by antiviral antibodies. But if I take the RBCs and we allow them to interact with antigens, you mix them with specific antibodies, then also it will form clumps. This will also be called as hemagglutination, but this is called as passive hemagglutination. And these kind of RBCs are called as passive carriers. So that means if I want to do a hemagglutination test, I take the RBC, the antigen is present on the surface of the red blood cells, and these antigens are generally polysaccharides. In that, when I see the reaction directly, that is the active test, but when I see the antibodies and take a sensitizing antigen, allowing it to agglutinate, then I call it an antigen sensitized RBC. And what is the kind of reaction I see? For any immunological method, we do not know exactly what is the quantity of antigen or antibody required to have a complete reaction. So what we generally do is put up a series of dilutions. If you look here, 2, 4, 8, 16, 32. So it is a serial dilution which has been put in to see and optimize at what concentration we are going to get a reaction, because a reaction can easily be masked by over antigens or over antibodies. So when I say the antibody has to have, it should be divalent, or there should be a multivalent antigen on the basis of which I am going to make the test. So it is very important to understand what is being formed. That means the antibody, we know the structure it is Y-shaped. So there are two parts which are the Fab parts, which are going to have the various sets of amino acids, which is the variable region, and we have the FC part. So when I look at the triangular antibody, what do I see? That it interacts and forms a lattice with the antigen, and this antigen has a multivalent antigenicity. That means it has multiple places at which the reaction can take place with the antibody. So it is bound to one antibody, the other antigen is bound to the other antibody, but please remember that it is the same site of recognition by the antibody. One antibody will recognize the same antigenic determinant present on the antibody. So present on the antigen. This is how a polyclonal antibody along with a multivalent antigen forms a lattice, which is the purpose of visualizing a precipitate reaction. And this there is a precipitation curve which was given by Heidelberger, and it clearly shows us that it is only at the point where the antigen and the antibody are equal will there be a lattice formation, and the side where there is excess of antibody, there will be no lattice formation, so there will be no precipitation and there will be nothing which is visual, which can be visualized. Similarly, if there is an antigen, a zone where there is an antigen excess, here also we are unable to form the lattice. So that is why the concentration of the antigen and the antibody have to be equal at the zone of equivalence. So that means what is the basis of this diagnostic method? It is a direct test. That means I can test the antigen directly from the sample, that means from the patient sample, or I can have an indirect test to detect antibodies. that is in the patient's serum. That means what am I looking at? Excess of the antibody once it is precipitated. It is the same curve, but how I am applying to a patient categorically tells me the lattice formation and the detection of the presence of antibody. When I say precipitation reactions, can I have soluble antigens? I am speaking of particulate antigens as well as I am speaking of soluble antigens. Soluble antigens and antibodies can easily be detected in a test tube. So if I look at the test tube, this is the blood, and this is the zone of equivalence where I have fed with excess antibodies. So what forms is called as a precipitin ring. The precipitin ring, which is visible, is also an area where the zone of equivalence is seen. So if I turn the curve and put it on this, it is exactly the same parabola where you have excess of antigens, the soluble antigens, excess of antibodies, again the soluble antibodies, and the zone of visible precipitate. So that means I'm talking about a precipitation test. This precipitation test is in the form of a solution. I can do a precipitation in other medium also, but this is directly in solution whereby I have the soluble antigen, I have the soluble antibody, and we allow it to interact. That means there will be a cross-linking present despite the medium. So can I have this test in a semi-solid medium? Was the question asked by the scientists. And yes, we do the latest agglutination, which is the pregnancy test, and this is done again not directly from the blood, but this is generally done by urine. So the urinary specimen also will give you agglutination. What does this mean? This means that this part of the experiment is different from the agglutination which we have just read about? No. It is again just the agglutination of the latex but not of the RBC. So that means what the latex particles have been coated with the human choriogonadotrophin hormone, and this has been done to test whether the person is pregnant or not. So I can easily differentiate it from the non-agglutinating latex beads. So this was a direct application of this test. When I talk of changing the medium, then I would not call it as active immunodiffusion. There is a process called as immunodiffusion. This immunodiffusion will involve passive immunodiffusion, and this passive immunodiffusion will be based on a concentration gradient. This concentration gradient will depend on the diffusion through the gelatin or the gel which has been prepared, and it is the network through which the antigen and antibodies have to pass. The diffusion will always depend on the electrical current, but the rate of diffusion of the particles will depend on the size of the particle, because if my gel which I have created is very concentrated, the particle which is of a larger size cannot go through it. So which is the size of the particle we want to diffuse will be based on the concentration of the gel. It depends on the temperature. It also depends on the viscosity of the gel. It depends on the hydration of the gel. And also, it will depend on the reaction of the matrix with the reactant. So, based on these diffusion techniques, they have devised a set of experiments which are based on immunodiffusion. They are the radial immunodiffusion, single or double diffusion. Double diffusion is called as Ouchterlony. Radial diffusion is based on a single diffusion method where the antibody is put into the gel and the known antigen is measured and put in a small hole which we create, and a precipitin ring is formed around the hole. So this is called a well. I would use that we cut a well in which we load the antigen and we allow the antibody and antigen to form a diffuse state in and around the well. When I say Ouchterlony double diffusion, that means I have both antigen and antibody in the wells in such a way that antigen diffuses from one well antibody diffuses from another well, and at some point of time they make a zone of equivalence. So my principle remains the same, but we have developed more techniques to identify and quantify whatever antigen unknown or antibody unknown we want to find. What is the medium we use? We can use agar, we can use agarose, and both. Agarose is nothing but purified agar. That means there should be no charge, there should be no interaction between gel and reactants, minimize them so that there is no cross reactivity. Agar is a high molecular weight polysaccharide from seaweed, but it has a very strong negative charge. So agarose, which is purified, is basically we have tried to minimize the charges to have clearer reactions. And agar is used to stabilize the diffusion powers which can permit us to visualize the bands correctly. So radial diffusion, antigen from the sample, antibody rich media, that means when I load the gel, the antibody is mixed with the gel and allowed to cool. Antigen and antibody will continue to react till there is a zone of equivalence which is reached, and we can easily measure the area. Look at the cartoon, you will be able to understand what I am trying to say. This is antibody containing the gel. This is the well which we have cut and we have loaded the antigen. Over a period of time, this is the ring which is going to form, and the ring will be formed only at the zone of equivalence. Zone of equivalence meaning equal amount of antigen versus equal amount of antibody. How do I interpret the data? That means you will take the diameter of the ring and we will directly proportional, it is directly proportional to the concentration of the antigen. That means, can I make it a quantitative assay? We can try to quantitate the antibodies based on this. Here we have a known antigen, so I have to identify the patient's antibody. So based on the known antigen, I can quantify the unknown antibody. How do I do it practically? This is called as the micrometer eyepiece, through which we look into it, and this is how we measure the precipitin ring. I'm sure it is visible to all the students. These are the markings, which are the standardized markings, and once we have taken the diameter, we can directly say that it is the extent of the ring gives you the concentration. So larger the precipitin ring, more is the concentration of the antibody, which we can quantitate with the known antigen. When I say double diffusion, what do I have? I have taken an antibody and I have loaded antigens, and we have allowed the precipitin ring to form. What do I see? I can easily see an arc, and this arc gives us the presence of a common epitope in the antibody. So that means this is a precipitin ring, this is the line which we have visualized at the zone of equivalence. When I see that the lines have crossed each other, that means it shows that there are two separate reactions which are occurring simultaneously, and there are no shared epitopes. So that means there are two antibodies, antigens having separate epitopes. What is an epitope? Epitope is the part of the antigen which is recognized by the antibody. But when I see that there is a spur formation, that means one line goes excess and the other stops, this is called as the zone or the line of partial identity. That means there are some part which is identifiable and the others are not. So Ouchterlony double diffusion method is also a commonly used clinical method, whereby we can understand the presence or absence of a particular antibody, and then we can move on to methodologies which are more sensitive and can be quantified in a finer way. Similarly, there is another method which is called as rocket electrophoresis, whereby the antibody is again loaded with the gel. So that means here I am running an agarose gel. I have cut the wells and put the antigen in the wells in different concentrations. We do to again identify the unknown antigen because we have excess of antibody and we have less of antigen. So what do we see? That we form the zone of equivalence in the form of rockets, and this categorically gives you the concentration. So my rocket, length of the rocket is directly proportional to the concentration. So as the concentration increases, the length of the rocket precipitin line increases. Well, that was talking about the soluble antigens. Now when I talk about agglutination reaction based on particulate antigens and antibodies, what do I understand? That there can be a direct agglutination on the cell, which is the direct agglutination, or attached to the latex beads, that is the passive agglutination. We have already gone through the pregnancy test, which tells us the passive agglutination. But when I say agglutination of a particulate matter, whereby I talk about the RBCs or about the bacteria or about the latex bead, what do I understand? That the blood group has also been based on agglutination reactions which are direct. That means the RBC cell surface should have antigens based on which there will be a reaction. Now, when I say RBCs, like I mentioned earlier, RBCs with the viruses will directly give you hemagglutination. That itself is called as the direct hemagglutination. But when I talk of ABO blood group, I am sure all of you have an idea that Karl Landsteiner was the pioneer who discovered the ABO blood group system in 1901. He and 5 of his co-workers started mixing their own blood, each other's blood, and separated the serum, mixed that, and kept on doing reverse, anti-reverse ABO groupings. What rule did he come to? He came to these 3 basic rules, that individuals will form antibodies to ABO blood group antigens they do not possess. Substances are present in nature which are so similar to blood group antigens that result in constant production of antibodies to blood group antigens which they do not possess. Very interesting. The third observation was critical for understanding compatibility between ABO blood groups. So, ruled by these 3 rules, we came up with the clinical significance of the observations of Landsteiner. We all know that immunizations are frequently done to protect us from diseases. We receive hepatitis immunization, we receive direct subunits of hepatitis viruses which are injected to raise the antibody titers, and the body recognizes foreign bodies and produces the antibodies. We also have been exposed to real hepatitis viruses which may result in the destruction of the virus itself. But ABO blood group also has antibodies which are again made as an immune response, but this does not result in destroying the cells. But in case we put a blood group which is not compatible with the patient, it results in hemolysis and kills the patient immediately. So that means there is something about inheritance about this immune group. Yes, these are the blood group antigens are codominant. That means if the gene is inherited, it will be expressed. But there are aberrant genotypes which do occur, but this is very rare. So Bombay antigen, H antigen is not part of today's lecture. But what do we understand? The basic understanding of genetics should be that there are 2 genes which are inherited from parents. Individual which has A or B is homozygous or heterozygous for an antigen. So heterozygous is AO or BO. Please remember, O is not another blood group. O just means null. That means it does not exist. Homozygous is when it is AA or BB. That means you have inherited A antigen from both the parents. Here, B antigens from both the parents. So phenotype is the actual expression of the genotype. That is, what will be the phenotype? A. Here also it is heterozygous, A and O. So what will be the phenotype? It will again be A, because O does not mean anything. When I say genotype, genotype will be AO in the heterozygous case, but AA in the case of homozygous. So when I do an example of determining the genotype, what do I say? That if there is B and O, A and O, AB is 25% chances that the child will be AB, or most likely 25% will be AO. In case it is B from the father and A from the mother, there is a chance of 25% chance of BO, and blood group O will be 25% chance. That means 1/4 population born have the chance to become AB, AO, O or B. But other examples could be, if like mom is AA and dad is BB, that means they are homozygous, 100% of the offspring will be AB. But if it is BO and OO, 50% will be B or 50% will be O. But if it is O, again 100% is going to have no antibody A or B. And if it is OO, it is going to be 50% of A or O. So if I look at blood group O, approximately 45% of the population is group O. No antigens for A or B are present. So this is called as a negative. O means there are no antigens present on the RBC, so no antibodies. And we shall continue later after a break. Okay, welcome back. As we were talking about blood groups, I was discussing about group O, whereby 45% of the population is group O, which has neither A antigen nor B antigen. But please remember, O is the American null, that means equivalent to 0. These individuals, they form potent anti-A and anti-B antibodies which circulate in the blood plasma all the time. When I say group A, that means 40% of the population is containing group A. There will be no B antigens present. Again, I repeat, all the antigens are present on the surface of the RBC, and these are polysaccharide in nature. So we always call them as cell surface antigens or RBC surface antigens. They are containing potent anti-B antibodies. So that means the blood will contain anti-B antibodies, but it will represent A blood group. It is raised against A. Group B, when I say the person belongs to B group, what does it mean? It means that the surface of the RBC contains the antigens which are B. The surface antigen is B. What is the circulating plasma antibodies? They will be anti-A. That means, is there a certain concentration of blood group B present in the population? Only 11%. When I say group AB, they are just 4%. And they contain both A and B antigens on the surface of the RBC. So it has neither A nor B. That means, what am I showing? That the surface antigen, although here the diagram has an error, it is showing just one antigen, it is supposed to show two antigens. So that means they possess no ABO antibodies. What happens if I give a different blood group to them? The moment an incorrect or incompatible blood group is transfused, there is hemolysis occurring because of the presence of antibodies which are opposing the antigens. And what results? The breakdown of RBCs. When I say AA, when we do the blood testing in the clinics, in the pathology lab, what do? We have antibodies which are circulating in the plasma, which is anti-B. What do I transfuse it with? I'm going to transfuse it with A group or O. When I have a blood group B, I will do it with B. Why? Because the antibodies which are there are A. So they will be compatible with the B antibodies. Similarly, AB, A and B or none. So that means, what do I understand? When I do a testing in the pathology, I don't do it with an anti-B, I do it with an anti-B. So that means there is an antibody raised against that. So it becomes compatible with the antigen of the blood. But whenever we do a blood grouping, we also look at another factor, which is the Rh factor, or we call it as a D antigen. This is also very important because the Rh, although we call it Rh, it is just because it was found in the rhesus monkey for the first time. That is why it is called as Rh. Whether it is a blood group system where we have to understand that there are many antigens, one is D, yes, we must understand it, but this also refers to just the presence of it. The absence just means negative. It is either present or it is absent, but there is no genetic inheritance of not having it. So that means, unlike the ABO blood systems, the people who lack D antigen cannot make it. They just do not have it. So production of an antibody to D requires exposure of the antigen to the antigen. That means the D antigen is immunogenic. That is, individuals exposed to it very likely will make antibodies. For this reason, all the individuals which are typed for D, if negative, must receive an Rh negative blood. They cannot take an Rh positive blood. So that means, if there is a female carrying a child, she must particularly be aware of the Rh factor, because if you immunize Rh and D antigen, the antibody can cross the placenta. Please remember, there are different subclasses of antibody, IgM, IgG, and it is IgG which is capable of crossing the placenta. And it is capable of destroying the fetus, which we have always seen in a cartoon, which I'm going to just show up, that any patient which has Rh negative is given Rogam, and that saves the child. That means, just consider, this is just the hemolytic disease of a newborn. A child is Rh positive. But during pregnancy, the fetal Rh positive RBCs, they will escape into the maternal circulation. The mother will produce antibodies to Rh, that means the positive, and the mother is negative. What happens? In the second pregnancy, the positive child results in destruction of all the fetal D positive RBCs. As a result, what does the patient require? The patient requires sensitization, and that is done by ROGAM. So that means this is part 1 of the reaction. Part 2, what I am talking now about, neutralization reactions. That means I can use antibodies to neutralize the impact of the antigens which have entered the body and which can cause destruction. Like we mentioned, it is very easy to detect a viral hemagglutination reaction because it happens as a soluble mediated reaction. In this, what do? We actually require the patient's serum and we can see the test. How can we counter it? That you take the antibodies which are antiviral antibodies and you can mix it with the cell. What do we see? That the virus and the antibodies will interact with each other and the RBCs do not get hemagglutinated. So this is a good neutralization reaction based on which we can save the patient. What do I understand by titers? When I say quantitate, what am I doing? I'm trying to observe and see whether the concentration can be determined or it can be quantified against a particular antigen. So when I do a dilution, it is either 1:20, 40, 1:80. That means this is a serial dilution and we are just trying to see the reaction. This is the top view of the well and this is the cartoon of or understanding of what happens inside the well when it is non-coagulated or agglutinated or it is agglutinated. Similarly, I also mentioned that there is a complement system which exists and there are complement assays which can also help neutralize the antibodies. That means if I have a CH50 antibody mediated hemolysis of RBCs by the complement, then what do? I can have functional assay that can determine the activity of the individual components or complement components, or we can have immunoassays which are again antibodies to the complement components and then the determination can further be made. What do I mean by all this? What I mean is you take an antigen and you allow it to mix with the complement and what do we see? That we see that there is a serum which will show the antibody against the antigen. This is called, and then you fix it. This is called as complement fixation. But if I have an antigen, you take the complement and there is serum which does not have the antibody, there will be no complement fixation. So this gives us an idea whether the presence of the antibody is present or not. It is not a theoretical thought. Clinically, what do we do with it? You take sheep RBC and you have an antibody to sheep RBC. You allow it to mix with the complement. And if there are no antibodies, you will see an antibody-antigen reaction, but the sheep RBC will not lyse. But you have the sheep RBC, you take the antibody, and there is combined complement available. What will happen? It will show agglutination. When we say there is a positive test, what do I mean? That all complement is fixed by the antigen-antibody reaction, and that when there is no hemolysis, then we call it as a complement fixation. But if an hemolysis occurs, I call it a negative test because there is no antigen-antibody complex which is present, so there is no antigen-antibody reaction which is occurring. So what is my indicator? My indicator is the presence of hemolysis or absence of hemolysis. And absence of hemolysis is what we require and consider as a positive test, not the hemolysis. Earlier we were taking hemolysis as the direct test where we were looking at hemagglutination based on the viruses. That was a direct test of the hemagglutination. So for a complement fixation, hemolysis is a negative test and no hemolysis is a positive test. Similarly, now techniques have improvised, and we have visualization techniques based on fluorescence. So we have a fluorescently labeled antibody technique which is direct. What do we take? You take the bacteria from the patient's throat. Here in case we have taken the example of streptococci, and you take a fluorescently dye labeled antibody to streptococci. When they make a reaction, the streptococci which are present will appear as a fluorescent. So directly we can visualize how many streptococci are present, and this is generally an indicator of a throat infection which can be quantified directly. There are also fluorescently labeled antibody techniques which are indirect. That means here again you take the T. palladium from the laboratory. You have specific antibodies in the serum of the patient. You interact this. You do not give this to the patient. All this is being done as a pathological test outside. So what you have done is only collected the blood of the patient. Then you again incubate it with the fluorescent dye. You label it with the anti-human immune serum globulin, and this will react with the fluorescent dye and the T. palladium, and what do we see? We see a big complex being formed which is going to fluoresce, and the fluorescence will be proportional to the amount of infection. So this is a positive test where the fluorescence is being seen, but this is indirect because you have taken antibodies raised against that the microbe, and you are incubating it with the fluorescent labeled dye, the antibody. So there are two antibodies involved, hence this is called as an indirect test. So how can I summarize these? I can summarize it in such a way that there is a precipitin reaction, and precipitin reactions are basically based on soluble antigens. Then we have agglutination, which are based on particulate antigens, that means the RBCs or the bacteria. Then I have hemagglutination, which I can specifically put under agglutination, and it is again the RBCs which agglutinate. We do not call it as aggregation. We always call it as an agglutination reaction. Then we have neutralization, that is, it is going to inactivate the toxins or the antibody causing viruses or bacteria. Then we have the fluorescent antibody techniques, and these antibody techniques are linked to fluorescent dyes. Then we have taken complement fixation, where RBC lysis is the indicator. That means if there is no lysis, then there is a positive test, and if there is lysis, it is a negative test. Then we have the ELISA, which is the enzyme-linked antibody, which actually can be very sensitive, and it can amplify both visibility as well as sensitivity. So I will continue with the common immunological assays, where we have ELISA, ELISPOT, flow cytometry, immunofluorescence staining, radioimmunoassay, and chemiluminescence. But today I shall be discussing only enzyme-linked immunosorbent assay, which is ELISA, and this is a very versatile method for detecting and quantifying a specific protein in a complex mixture. This was initially given by Engvall and Perlman in 1971, and this enables analysis of protein samples which can be immobilized in microplates using specific antibodies. The technique has revolutionized immunology and is commonly used in medical research laboratories. ELISA also has a lot of commercial applications, including detection of disease markers, allergens in diagnostic and food industries. So what is an ELISA? Like I mentioned, that there is antigen. I can quantify the antigen, so I can detect the level of an antibody or an antigen in a sample. What are the basic elements? That means I can have a capture ELISA, direct or indirect immobilization of antigens to the surface of any polystyrene microtiter plate wells, or I can have a blocking, where that means I do an addition of irrelevant protein or other molecule to cover up all the unsaturated surface binding sites of the microplate. Or I can have a probing or a detection based ELISA, where I incubate with the antigen specific antibody. that affinity binds to the antigens. Or I can have a single measurement, that means detection of the signal which is generated via direct or secondary tag on the specific antibody. So based on 4 elements, I am going to move on to other tests. So what are the types of ELISA? Indirect, sandwich ELISA, or competitive ELISA. So when I look at indirect ELISA, what do I quantify? I quantify antibody, and what is immobilized? An antigen is immobilized here. So you take an antigen, immobilize it on the microtiter plate. You put a primary antibody. This antibody saturates, remove the excess, add a secondary antibody which is conjugated with an enzyme, incubate it, remove the excess conjugated secondary antibody, then put a substrate. So this substrate is chromogenic, and this leads to a colored reaction, a color which can easily be read by optical density measures. And what we read is the colored reaction, which can directly tell us how much of antibody which is bound. So directly, indirectly, because it is using 2 antibodies, it gives us the quantification of the antigen. So when I say there is a sandwich ELISA, hereby I will test an unknown antigen. So you coat the microtiter plate with a known antibody, which is called as the capture antibody. Then you incubate with the unknown antigen from the patient, and then you again use a primary antibody to saturate. It will bind to the antigen. Use a secondary antibody, which is going to have an enzyme linked to it, incubate it with the substrate, and you get a colored reaction, which is again proportional to the color. The concentration can easily be assessed based on that. The third type is the competitive binding immunoassay. Hereby, what do? Again, I have an immobilized antibody. And this immobilized antibody, you saturate with the antigen, and you allow the enzyme linked antibody, and it competes for the same site. So when I use another antigen, it is going to compete for the same site, and based on that there is a competition, and we can easily measure the amount of enzyme linked with the chromogen substrate. So my color reaction will be not directly proportional but inversely proportional. So what are the enzymes and substrates I use? Horseradish peroxidase is an enzyme which is generally used, but it requires 3 kinds of substrates can be used. OPD, that is O-phenyl N-diamine dichloride, and the absorbance is read at 492. We have TMB, which is tetramethylbenzidine. It is read at 450 nanometers. Or if I have ABTS, which is, as I know, bis 3-ethyl benzothiazolene 6-sulfonic acid, and this is a diammonium salt. This can be read at a maximum absorbance of 410 nanometers. There are more enzymes. I can have alkaline phosphatase as an enzyme. For this, the substrate required would be PNPP, which is p-nitrophenyl phosphate, and this is a disodium salt. This can be read at 405. So what do we see? That between 405 and 490, we can read most of the chromogen assays which are based on enzymes. This is the optical density which we read. We make a standard curve, and we can use the optical density or the OD which we read in arbitrary concentrations, or the units are arbitrary, and this gives us an idea of the quantification of the enzyme linked assays, which are very sensitive. Based on this, they have come up with another assay which is called as ELISPOT, and ELISPOT is an improvement on an ELISA. Here we can actually see a spot. The principle is same. The principle is based on the sandwich ELISA. If you remember, you can recollect, sandwich ELISA was where the antibody was immobilized, the antigen was present, and then there was another antibody which was capture antibody, and then primary antibody with a secondary antibody. So why do I need to do an ELISPOT when I have already understood what is a sandwich ELISA? Please remember that is done in a soluble form. Here we can actually identify the cells which are secreting cytokines or growth factors, and wherever the cell is secreting, a spot is formed. So you can count the number of cells which are secreting these cytokines. So as a result, the quantification is not of how much of enzyme is present, but how many cytokine secreting cells we have in the plate also can be assayed. Now, if you look at the cartoon, it is very easily understood. You take the antigen, you incubate it with an antigen secreting cell, but my well has to be antibody coated. So that means the cell is secreting the antigen, the antibody has to recognize it. Then very carefully we can remove the cells by washing. The secreted annulate is captured by the immobilized antibody. You can see that the antigens which were secreted we have bound to the antibody. You can then incubate it with a biotinylated, that means it is an antibody which has been basically labeled with biotin. You incubate it with an alkaline phosphatase conjugated streptavidin, because biotin will combine with streptavidin. And when you look at the streptavidin, what do we see? That you can actually form a black spot, where whenever we do the addition of the substrate, and we can see the formation of the spots. So what do I have? That these many cells, that this cell which is present here has secreted so many cytokines. So that means now we have an understanding of the population of the cells that are secreting the cytokines or the growth hormones. So we can also have certain labels, like we have used as enzyme as a label, and what are the most commonly used labels are radioactive labels, which I use for radioimmunoassay. We have enzymes, we have fluorescence, and we have chemiluminescence. We can further move on to a radioimmunoassay which is based on competitive binding. That means I use radioactively labeled iodine as a label, and this competes with the patient for the site of the antibody where it is bound to the iodine. What is the purpose? High radioactivity, small amount of patient substance, or if there is low radioactivity, high amount of patient substance. So when I say radioimmunoassay, why do we want to use this? This is very sensitive. It is highly sensitive as compared to ELISA, and it can be used to measure very small concentration of antigens. Here the known quantity of antigen is generally made radioactivity by labeling it with iodine, and this known antigen and the patient sample are added to the reagent antibody simultaneously. So the known antigen means the radioactively labeled antigen will compete with the site for the patient's antibody, and it is going to compete for the same site on the antibody. So the antigens can be separated from the unknown ones. How? As it leaves the antigen, we can keep on measuring the amount of radioactivity which has become unbound, because you have already saturated the antibody with the known antigen. So as the known antigen moves away, we can measure the radioactivity and make an assessment of how many antigens competed and the unknown got bound and the unknown one was released. So here the results are inversely related to the label detected, because the label is leaving, not adhering to it. And we always run the standards to actually make a standard curve so that we can plot it against it to know the exact quantity. Let us understand what is competitive binding. That means I take a known antigen and you take an unknown antigen. Allow it to compete for the same site on the antibody. But please remember to wash off the unreacted substances. This will determine the method of detecting. That means I have a radio label and I have a non-radio label. This is my antibody. you go on adding excess of antigen and it is bound to compete, because they do not understand whether it is radio-labeled or not. It is just the excess of antigen which basically leads it to leaving the antigen. In a non-competitive binding, the patient, you take the antigen and antibody, and depending on what is being tested, I can directly measure the detection. What is the standard? When I say there is substance which you know the concentration, that will form a standard. And we already run from a low to a higher level so that we can create a standard curve at all levels. So, what have I taken? The labeled antigen. We have allowed it to incubate with the antibody. So, the antibody is completely saturated with the label. When I test the patient sample, this is unknown, that is, the antibody does not make a difference between the labeled antigen and the non-labeled antigen, and it will go on binding to the unknown antigen. As a result, the labeled antigen moves away, and as it moves away, we can go on detecting the radioactivity by means of machines. So, this is again an assay similar. So, when I say competitive, I can again have a latex bead assay whereby we understand. And to understand the whole immunoassay, we do a radioactivity, same thing what I have explained earlier, but this is the complete assay where we can know the difference between the unknown and the known. Thank you.
On that note, I would like to thank ma'am for this very enriching discussion, and I hope, dear friends, you have come to understand what immunological techniques are all about. On that note, thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.